And our final speaker this session will be Jianan Gong. She's in the Cancer and Haematology Division, and she works a lot with David Wang and Andrew Roberts. And she's going to talk about targeting multiple myeloma with BH3 mimetics.
Firstly, I want to thank the organizer for this great opportunity to present our work. So today, I'm going to talk about targeting the pro-survival BCL2 proteins to treat multiple myeloma. So what is multiple myeloma? So it is a cancer of plasma cell, which is a type of white blood cell normally responsible for producing large amount of antibody to protect us from pathogens. So for this disease, it can affect multiple organs, but one of the typical phenotype is the accumulation of these malignant plasma cell within the bone marrow in the patients. So multiple myeloma takes about 10% of blood cancers and 1% of all cancers. It is considered treatable but generally incurable. So current treatment for the myeloma patients include corticosteroids such as dexamethasone, proteasome inhibitors such as bortezomib, and the immunomodulatory drugs such as lenalidomide or pomalidomide. And the myeloma cell is characterized by very complex cytogenetic and molecular aberrations. So as the previous speakers has introduced a lot of background information about their BCL2 protein and their relationship with a lot of disease, including cancer, so I will jump into the topic today directly. So the question we are interested in here today is, can we target those pro-survival BCL2 proteins to treat, to kill myeloma cells? So also, as you have heard a lot from the previous speakers, that now we have multiple approaches to target these pro-survival BCL2 proteins. Firstly, we have a panel of these B3 mimetic compounds, which are small B3 molecular compounds to target these different pro-survival BCL2 proteins. Just remind you very quickly that the ABT737 can target BCL2, BCXL, BCLW, and we also have a BCL2 selective inhibitor and BCXL selective inhibitor. As you have heard from Gemma, that recently we also have a very good MCL1 selective inhibitor. So another approach, very efficient approach that we can use to target those pro-survival BCL2 proteins is to use the CRISPR-Cas9 technology, as you also have heard from Gemma, that to delete these pro-survival genes at the genetic level to study their function. For example, we can use a CRISPR to delete MCL1 specifically in the myeloma cells to see how the cell responds to the loss of MCL1. So the first question we ask it is, how do these myeloma cells respond to these B3 mimetic compounds? So in order to answer this question, we collected 27 myeloma cell lines, and then we treat them with different concentration, with serial diluted concentration of these B3 mimetic compounds, and then we test the cell viability at 48 hours. As you can see that, of these 27 myeloma cell lines we tested, about 4 of them are highly sensitive to the BCL2 selective inhibitor with IC50 below 500 nanomolar. As you have heard from Gemma, that IC50 represents the drug concentration that can kill 50% of the cell. And we have a distinct subgroup of myeloma cells highly sensitive to the BCXL selective inhibitor. But as you can see from the whole Venn diagram, that the majority of these myeloma cells cannot be killed by either targeting BCL2 or BCXL. So the question now is, we ask it is whether MCL1 might play a role here. So the reason we are interested in MCL1 rather than other pro-survival BCL2 proteins such as BCLW or A1, as you have heard from Gemma, to that the MCL1 play a very important role in multiple cancer cells. And also there is a study which is also from WEHI to look at the role of MCL1 in the normal plasma cell. So as you can see here, that the white column represents the mice with a wild-type MCL1, and the green column represents the mice with one copy loss of MCL1, and the both copy of MCL1 were deleted was shown in black column. And then they look at the loss of MCL1 on the level of naive B cell or plasma cell in the spleen or bone marrow. As you can see from the black column that the deletion of MCL1 can lead to dramatic decreased level in both of plasma cell in both the spleen and bone marrow, and the drop of plasma cell is much more significant than that observed in the naive B cell. So which suggested to us that MCL1 is essential for the survival of normal plasma cells. So what about these malignant myeloma cells? So let's go back to this Venn diagram. So as I have shown you earlier that we tested 27 myeloma cell lines, and of these 27 myeloma cell lines, a small proportion of them are sensitive to either the BCL2 or BCL inhibition. So what about how they respond to the MCL1 selective inhibitor? So surprisingly, we found of these 27 lines we tested, 25 of them are very sensitive to the MCL1 selective inhibitor, which is really encouraging. But there's still a question mark here is that whether this MCL1 inhibitor kills the cancer cells through its on-target activity or its non-specific effect. So in order to answer this question, so the next thing we did is that we use the same CRISPR-Cas9 technology to generate a panel of back-to-back double knockout myeloma cell lines, and then we treat them with the MCL1 inhibitor to see whether the loss of back can protect the kidney by the drug. So AM01 is one of the most sensitive cell lines to the MCL1 inhibitor. As you can see, the y-axis represents the percentage of a live cell, and the x-axis represents different concentration of drug. As you can see from the black line that the wild-type AM01 cell is super sensitive to the drug. However, the deletion of back can protect the kidney by the drug completely. And we have the similar observation in another two myeloma cell line, H929 and KMS12BE. But you can see in the H929, at the highest concentration of drug, there's slight off-target effect. So I have introduced you in the very beginning that there's another approach that we can use to target the gene to study their function is to use a CRISPR-Cas9 technology. So today I will just show you one example with OPM2 myeloma cell line. So this is a drug treating data. As you can see from the blue line that the OPM2 cell is very sensitive to the MCL1 selective inhibitor, but not that sensitive to the BCL2 or BCL inhibition. Consistently, when we use the CRISPR-Cas9 technology to delete these pro-survival genes at the genetic level, only the deletion of MCL1 can lead to dramatic drop of cell viability, which further validates that MCL1 is a critical survival factor for these myeloma cells we tested. So another thing I mentioned in the very beginning is that we know the myeloma cell is characterized by very complex cytogenetic and molecular aberrations. So another thing the next thing we did is we look into this data to see whether the sensitivity to the MCL1 inhibitor is related to certain subtype of myeloma cells. So in order to do that, we group these myeloma cells into different subgroups in terms of their chromosome translocations they carry. As you can see here, each dot here represents one myeloma cells, and the y-axis represents their IC50 to the drug. As you can see, there's no clear relation. The sensitivity to the MCL1 inhibitor is not restricted to certain subgroup of myeloma cells. And more importantly, the myeloma cells which harbor the chromosome 4 and 14 translocation, which we know in clinic that they are associated with poor prognosis, can also be killed by the MCL1 inhibitor as well. So at the same time, we also look at the TP53 status to see whether the sensitivity to the MCL1 inhibitor is dependent on their TP53 status. As you can see, even the myeloma cells with a mutant TP53 can be readily killed by the MCL1 inhibitor as well. So until now, we can conclude that targeting the MCL1 inhibitor in vitro can kill the majority of the myeloma cells we tested. So I think the next thing we are very keen to know is that whether the drug is also active in vivo, whether it can also kill the myeloma cell in vivo. So in order to answer this question, we established two myeloma xenograft models. One is to inject the tumor cell into the mice subcutaneously so that the tumor cell can grow under the skin of these mice. And then at day 10, we will divide these mice into two cohorts. Half of them will be treated with vehicle, and the other will be treated with MCL1 inhibitor. As you can see, the y-axis represents the tumor volume, and the black line represents the mice treated with vehicle, and the treatment with MCL1 inhibitor can delay the tumor growth. However, there's a very big disadvantage of this myeloma xenograft model is that the tumor cell grow as a bump under the skin of these mice instead of in the hematopoietic system, which is a myeloma cell should grow in. So the next thing we did is that we established the other myeloma xenograft model. So instead of inject the tumor cells into the mice subcutaneously, we inoculate the tumor cells by IV injection to see whether the tumor cell can grow in the hematopoietic system. So this is to show you the result in the peripheral blood. So the y-axis represents the absolute number of tumor cells circulating in the blood. So the first information we can get is that, and then we observe the tumor cell in the blood at different time points. So there's one information we can get that, firstly, the tumor cell indeed can grow in the peripheral blood when we inject the tumor by IV injection. And the other information we can get is that as the disease progresses, you can see that there is more tumor cells circulating in the peripheral blood, which we can use to monitor the tumor burden. So after we establish this mouse model, so the next thing we want to know is how the MCL1 inhibitor responds in this IV injected mouse model. So this is to show you the survival data to see whether the treatment of MCL1 can prolong the survival in the IV injected mice model. So whenever the mice is alive, it will be shown at 100%. And whenever the mice get sick, the line will drop. As you can see, compared to the black line, which is treated with vehicle, the treatment with MCL1 inhibitor can prolong the survival by 11 days. However, the response is not as good as we expected in the subcutaneous model or in the model as JAMA as shown in the lymphoma. So I think there are a few possibilities. Firstly, it's inadequate MCL1 inhibition, and the second possibility is that inhibiting MCL1 alone is not sufficient to kill myeloma cells in the hematopoietic system. So in terms of the first possibility, we are now testing different drug dose and schedule of MCL1 inhibitor to see whether we can improve the efficacy. So for the following talk, I will mainly focus on the second possibility, trying to ask the question whether limiting the activity of other pro-survival BCL2 protein can improve the response to the myeloma cell to the MCL1 inhibition. So as our in vitro mechanism data suggested that, which I don't have to show you today, suggests that BCL2 is another very promising candidate. So for today, I will just show you what we get for now. So in order to do that, we use the CRISPR-Cas9 to get the BCL2 knockout AM01 cells, and then we inject these myeloma cells into the mice, also in IV injection, and then we did the same treatment to see whether the deletion of BCL2 can prolong the response, can enhance the response to MCL1 inhibitor in the IV-injected mouse model. As you can see here, that the survival date is doubled to 22 days when the BCL2 is deleted in the AM1 cell. So to summarize my talk today, firstly, our mechanism study showed that the MCL1 inhibitor is killing tumor cells through its on-target activity, which is mediated by the back-mediated apoptotic pathway, and targeting MCL1 kills most myeloma cells tested in vitro. Other pro-survival BCL2 proteins, such as BCL2L, BCLXL, play a role in maintaining some of the lines we tested. However, targeting MCL1 alone might not be sufficient to treat myeloma cells in vivo. So our very early data suggests that targeting other BCL2 proteins, such as BCL2 itself, might be used to enhance the response to MCL1 inhibition in the myeloma. Finally, I would like to thank my supervisor, David and Andrew, for their great support and mentor, and all the other members in our lab, especially David Siegel for his great help as always, and all the people involved in the MCL1 inhibitor study, especially Olivia and Gian, and the CRISPR-Cas9 and Marco, who established the CRISPR-Cas9 technology, and all the facilities in WEHI on fundings for supporting this work. Thank you.
I was wondering, is it feasible to somehow test growth in the bones?
Oh, that is the other thing actually we did, but we didn't show in the slides today. So we also test the percentage and the absolute number of tumor cells in the bone marrow after we treat the mice with MCL1 inhibitor, and it does show that there's a reduction of tumor cell in the bone marrow.
There's a reduction, but not as good as in the blood.
Yeah.
Any other questions? Thank you.
Thank you.